Welcome to Wildcat Science with Mrs. Haugo. This is Human Biology Genetics, and we are in video 2, part 2. Today we're going to be starting out talking about dominant and recessive genes. So, please write this down. Dominant genes will express their effect regardless of any other allele. So if there's a recessive gene, it doesn't matter. The dominant gene will be the trait that shows. You only need one allele to show this trait. So let's say you're homozygous for a trait. Homo means same, so then you would have two of the same alleles when both alleles for a trait are the same. So example, you could be homozygous dominant, and remember dominant is capital letters, so capital R, or you could be homozygous recessive, which would be little r. So the recessive trait only appears when you have two of them. So you have to have both recessive alleles to show a recessive trait. So for example, plant with genotype r will have wrinkled seeds because there is no dominant round seed allele. An organism could be heterozygous for a trait, and that's when there are two alleles that are different. So a capital R and a little r. So if you have capital R for round and little r for wrinkled, what will the plant look like? Well, it will have round seeds because the dominant allele expresses the trait. Okay, phenotype is what you see. It's the physical characteristic. Any characteristic that can be observed or tested. So some examples, seed shape, eye color, height of a person, blood type, or being able or unable to taste this PTC compound, which your book talks about on page 561. And the genotype is the genetic makeup. So the capital R, little r, big R. Okay, so circling back to our PTC compound on page 561 in our book in chapter 25. PTC stands for phenothiocarbamide. And it is a bitter compound that some people can taste and some people cannot taste at all. And it is, the ability to taste it is a dominant trait and it's passed on genetically. So if you get one allele for tasting this bitter compound, you'll be able to taste it. And if you get two recessive alleles, then you will not be able to taste it. So the genotype is your, which two alleles you get. And then the phenotype is like if you were to taste it or not taste it. Okay, so this got cut off, so let's take a look at that. So big R, little r, XY, XX, those are all genotypes. Okay, so the distribution of chromosomes to offspring. So reproductive cells, the egg and the sperm, are produced by meiosis. During the meiosis process, the number of chromosomes is halved, so the reproductive cells have 23 chromosomes. So all of your cells, except for egg and sperm, have 46 chromosomes. And then it's important that your egg and sperm only have 23, so when an egg and sperm come together, then they have the regular 46. Okay, we're going to review the steps of meiosis, so please write these down. Interphase. Cells prepared to divide and chromosomes replicate. We're going to look at a simulation in class. Then the next step is prophase 1. The chromatin condenses into chromosomes, the nuclear membrane disintegrates, and the centrioles and spindle fibers appear. An important thing that happens in prophase 1 that doesn't happen anywhere else is that crossing over can occur. So those paired chromosomes, we're going to call them homologous pairs, line up and they can switch little sections with each other. So what is crossing over? An exchange of alleles between paired chromosomes, and that results in more variation in offspring. So maybe you had a capital A, capital B, capital C, little a, little b, little c, and then we switched, so now you have capital A, capital B, little c. So you have different variations. After prophase 1 is metaphase 1. So the spindle fibers go from the centrioles to the centromeres, which is the middle of each chromosome. And those paired chromosomes get lined up at the middle of the cell next to each other. And then after that is anaphase 1, where the spindle fibers shorten and they pull the chromosomes apart. So in this one, the paired homologous pairs split apart. The sister chromatids, the replicated chromosomes, don't split apart yet. So in anaphase 1, the homologous pairs split apart, are separated to opposite ends of the cell. Like I said, those sister chromatids stay together. After anaphase 1 is telophase 1. The nuclear membrane reforms, your chromosomes unwind, the spindle fibers and the centrioles disintegrate, and there's cytokinesis. That one cell splits into two. So now each daughter cell contains the same number and same type of chromosomes, but the cells are genetically different from each other, and they're genetically different from the original cell. Now we're going to do it all over again. Prophase 2. The nuclear membrane disintegrates, the chromatin condenses into chromosomes, the centrioles and spindle fibers appear, but you notice there's no crossing over. That only happens in prophase 1. Metaphase 2, the chromosomes, now the sister chromatids, line up on the metaphase plate. So before they lined up with their homologous pair, but now they're lining up with their sister chromatids. Okay, anaphase 2. The spindle fibers shorten and the sister chromatids split and go to opposite sides of the cell. And in telophase 2, the nuclear membrane reforms and the chromosomes unwind into chromatin, and then there's cytokinesis. Each cell splits in half, making 4 cells that each have 23 chromosomes and are genetically different. Okay, so let's talk about Punnett squares. This is another tool for genetics. It's a tool used to show all the combination of genes that can result from a given parental cross. Given the genotype of the dad and the genotype of the mom, what could possibly be their offspring? So it shows all possible allele combinations, so the combinations of different alleles, and the theoretical ratios. So let's say theoretically it's a 1-to-1 ratio, or 1-to-4, or a 9-to-3-to-1 that the offspring will have these traits. Okay, we're going to do a little bit of practice in class. So there is some monohybrid and dihybrid practice, and we're going to watch a little video about Punnett squares and Mendelian genetics. So jumping into some different types of traits, there are sex-linked traits, which are found on the X chromosome. So these are traits, these are alleles that are on the X chromosome. Sex-linked recessive disorders are much more common in males. And I want you to think about why males would get a recessive disorder that's found on the X chromosome much more often than females. Some examples of sex-linked traits are hemophilia, colorblindness, and male pattern baldness, which are all more common in males. There's our why question. So a female who has 1 copy of the recessive gene are considered carriers. So you can pass the trait on, and so your girls would need 2 copies of the genes, and your boys would only need 1 copy of the recessive gene to show the trait, because they're going to get a Y And the Ys don't have as many chromosomes on them. They don't have, they don't have as many genes on them. So they don't have anything to mask it. So let's say you are a female with a colorblind father. So what would his genotype be? You marry a normal-sighted man. What percent of your sons will be colorblind? What percent of your daughters will be colorblind? So I want you to figure this out, and then we're going to talk about it in class. Let's talk about some different types of hereditary traits. So some hereditary traits are determined by a single gene. So some examples are a cleft chin, free versus attached earlobes, hitchhiker's thumb, which I do not have, a widow's peak, which is where your hair kind of comes down to a little V. I don't have that. Wet or dry earwax, cystic fibrosis, that disorder is a one disease, and sickle cell anemia is determined by a single gene. And there are many more, but most of your traits are not determined by one gene. Most of them are what called multifactorial inheritance, which means a bunch of different genes affect what trait that is. So hair color is more than one gene. Eye color is more than one gene. Height, intelligence, athletic ability, a lot of different things are affected by more than one gene. Also, your environment affects a lot of these more complicated traits as well. Skin color, and then another one we didn't mention was the genetic predisposition for heart disease is more than one gene. Okay, so let's look at simple dominance. So this would be, some traits are determined by simple dominant versus recessive alleles. So dark hair is dominant over blonde hair. Tongue rolling is dominant. Can you roll your tongue? Codominance. Both alleles are fully expressed. So if you have a, let's say, black and white feathers, if it's simple dominance, let's say black feathers are dominant over white feathers. So if you have two alleles, you'll just have a black feathered bird. If it's codominant, you would have a black and white feathered bird. Blood types are codominant. So if you have an A and a B, you have AB blood. So you don't have A blood or B blood or like A and a half blood. You have AB blood. Both of them are expressed. And then there's another type called incomplete dominance, where both alleles are expressed but not fully. Like, so you get, the trait you get is in between. So if you had black feathers and white feathers, you would get gray feathers. So another example is wavy hair is when curly hair and straight hair are both expressed. You get something in between. You get wavy hair. So let's talk about the interplay between genes and the environment. So there's a lot of things that are involved in both the environment and your genes. For example, intelligence, height, weight, lifespan, susceptibility to certain diseases. So let's go with one example. Person X with a dominant gene for facial freckles will show freckles after sun exposure. They have the genes and the environmental turn on, which is they don't get freckles in the winter, but once they are out in the sun in the summer, they will have freckles. Okay, another example, let's say person Y and Z both have genes associated with obesity. Person Y grows up in a home that cooks healthy food and loves active outdoor sports. They're not obese. Person Z grows up in a home with junk food all the time, no regular meal schedules, everybody spends a lot of time in front of the TV or on their phone. Person Z becomes obese. They both had the same genes, but the environment turned it on in one and not on in the other. So we're going to pause there. All right, thank you. Bye.